Hello everyone, thanks for coming to my talk entitled The Relationship of Cell Volume to Cell Health. The story that I'm going to tell today is a reflection of the many customers with whom that I've interacted with in R&D, process development, quality release, as well as my years as an assistant professor of cancer biology at the University of Chicago. The title of my talk today is The Relationship of Cell Volume to Cell Health. Cell volume is a critical parameter that can be used to understand cell health and cell state. Cell volume is a universal indicator of cell state that can provide insight into whether the health or state of cells is expected or out of range of what should be expected and should be further investigated. Cells often swell or shrink in response to toxic conditions or cell state changes. T cells have been shown to swell upon their activation, and this swelling is often an indicator of persistence of T cells. The volume changes are often very small, often femtoliters, and these changes are often too small to be observed by imaging or flow cytometry using either forward or side scattering methods. But the Coulter method can detect these small changes. The ability to precisely measure cell volume is therefore a powerful tool for cell biologists and immunologists alike. The objectives of my talk today are to describe the strengths and weaknesses of several contemporary cell volume analysis tools, specifically with regard to cell volume. I will describe several important historical examples where cell volume was a critical parameter describing the quality or state of cells. Finally, I'll describe a specific experiment that we performed to highlight the differential ability of the Coulter method, the flow cytometry method, and the automated imaging method for capturing acute changes in cell health. Three commonly used methods for cell volume and health determination are flow cytometry, 2D imaging, and the Coulter method. Flow cytometry is a method for cell analysis. By mixing cells with various dyes for different time periods, one can obtain many different parameters regarding cell health and state, including cell cycle, receptor expression, etc. And using this method, you can gate particular populations that are stained or not stained to understand the percentages of cells in a particular state, such as apoptosis, necrosis, or cell death. You can also interrogate the forward and side scattering profiles of cells in order to obtain a bit more information regarding cell size and granularity. A common assumption among flow users is that forward scatter is directly associated with cell size, and that side scatter is directly associated with cell granularity. In actuality, forward and side scatter are complex functions that depend upon many parameters, including laser wavelength, cell size, light scattering collection angle, and refractive index of the flow medium. In summary, using forward and side scatter as indicators of size and granularity is not straightforward, and doing so can often mislead the researcher regarding cell volume and granularity. 2D imaging is a relatively inexpensive method that is often used to visually determine cell size relative to reference marks on a hemocytometer using a microscope. However, the method is tedious to do by eye or by hand, and impractical for sizing and counting of hundreds of cells. Furthermore, determining 3D volume based on 2D images can be very complex depending upon the cell shape. Lastly, cell volume obtained from 2D images is not as sensitive to small changes in volume as the Coulter method, given that changes in cell area are a squared function of changes in cell radius, whereas changes in cell volume are a cubed function of cell radius. The Coulter method provides a very precise understanding of cell counts and cell volume. In order to employ the Coulter method, one simply places their cell suspensions in a cup containing an isotonic solution, and then places the cup in the instrument. The instrument then aspirates a defined volume of liquid through a small hole in a glass tube. Each time an object blocks the hole, a voltage pulse is measured that is proportional to the size of the object. The instrument is able to measure several thousand cells or other particles per second, and therefore provides very nice statistics of the size distribution of cells or other particles in the sample. Following a few seconds of analysis, one will then obtain data that can be displayed in the form of a histogram. In this example, whole blood was analyzed on the multisizer 4E. You can clearly observe one peak corresponding to red blood cells and another peak representing cells of higher volume, corresponding to white blood cells, monocytes, and other cells in the sample. Following red cell lysis, the smaller red cell fraction was eliminated in this example. When the histograms are overlaid, you can clearly observe the red cell fraction with a median size of 5.17 microns and the larger non-red cell fraction with a median cell size of 6.14 microns. Nearly 10 years ago, Bruce Levine's lab published work in which they used the Coulter method in concert with flow cytometry to monitor the fractionation of blood cell populations. Although the forward and side scattering profiles obtained with the flow cytometry method were complex to interpret, the Coulter cell volume analysis method provided clear evidence that fractions 2, 3, and 4, separated by their elutriation centrifuge, contained cells of different size. By combining the information from both the Coulter and flow cytometry method, Bruce's team was able to clearly see that fraction 4 contained T cells, B cells, and monocytes, whereas fractions 2 and 3 contained only T cells and B cells. In a subsequent study, Carl June's lab made an exciting discovery regarding the relationship of cell volume to T cell persistence. In this study, they made a series of chimeric antigen receptor variants which contained constant extracellular domains and a series of uniquely engineered intracellular regions along with GFP as an expression readout to monitor by flow cytometry. Following transduction of T cells with lentivirus harboring these CARs, the group stimulated the T cells with CD19 expressing K562 cells and then used flow cytometry to monitor expression of CARs on cell surface and measured cell counts by the Coulter method. They found that cells harboring the CAR containing the 4-1BB domain were more persistent in the proliferation capacity and were larger in volume than cells harboring the other receptors. Furthermore, the authors noted that cells harboring the 4-1BB domain had greater cancer-killing potential than cells harboring the other receptors. The authors also referenced one of their earlier publications noting that, indeed, electronic cell size analysis was the most reliable method to determine when to re-stimulate with additional immobilized monoclonal antibody. In essence, the authors of this work rediscovered the finding that active CAR T cells have a greater volume than inactive T cells, and that cell volume is a key indicator of T cell persistence. In this final part of my talk, I'll discuss an experiment that we performed to highlight the acute changes in cell health that can be observed by monitoring cell volume with the Coulter method versus other commonly used cell analysis methods. In this experiment, we treated Jurkat cells with 2 micromolar staurosporine and then tracked the viability of cells with a ViCell Trypan Blue automated cell analyzer, a Cytoflex flow cytometer, and lastly a multisizer 4E Coulter counter over a 6-hour period. We tracked Trypan Blue exclusion using the ViCell automated cell imaging instrument, and by this method, the cells showed no change in viability over the 6-hour period. The viability of untreated cells is shown in blue on the histogram, while the viability of treated cells is shown in orange. The images below demonstrate the levels of Trypan Blue exclusion from the cells at untreated time 0, as well as the treated time point following 6 hours. In summary, no significant change in viability percentage was observed during the time course. When analyzed by Yopro-1 and propydium iodide staining with a Cytoflex, Also, no substantial increase in apoptosis, necrosis, or cell death was observed during the 6-hour period in the treated versus the non-treated cells. When examined by the Coulter method, a significant change in cell volume is observed, and specifically the treated cell population in as little as 30 minutes. The treated cells begin to shrink and continue to get smaller throughout the 6-hour time period. In contrast, the volume of untreated cells, shown at the bottom, is very constant and it's unchanging over the period of the time course analysis. When each of the methods is overlaid, it is clear that examination of cell volume by the Coulter method provides very immediate awareness of cell health change versus analysis by trypan blue or flow cytometry readouts, whereas no significant change in cell viability or forward or side scattering can be observed when examined by the flow cytometry method or the ViCell automated imaging method. In summary, cell volume is a critical universal parameter that describes cell health and cell state. Cell volume is a useful way to discriminate cell types following elutriation or other separation methods. The volume changes that accompany cell health and state are often too small to be detected by flow cytometry or imaging methods, but are easily able to be captured by the Coulter principle method. CAR T cells have been shown to swell upon their activation, and their persistence in cancer killing is often marked by sustained increases in cell volume. The ability to precisely measure cell volume is therefore a powerful tool for cell biologists and immunologists who need to track the health and activation state of cells. If you would like further information about the multisizer or other Coulter products, please contact me or your local Beckman Coulter Life Sciences representative. Thank you very much for your attention.